Hi, everyone. Welcome to this week's Single Cell and Exploration Club. My name is Julie. I'm a scientist at BioTuring. And so for today's club, we will be characterizing microglia subtypes in Alzheimer's disease. So in this data set, I was able to identify some activated microglia subtype. And for this week's session, I will be giving a demo on both the BrowserX Pro and BioVinci. So one of the features that I want to spotlight here for BBrowserX Pro is our cell type prediction feature. So we have a lot of different tools built into the app to help you annotate your cell types. But for the demo today, I will be mostly going over the MetaReference. And so I'll be going into depth into that, how to interpret the results, and then how to also visualize them on BioVinci as well. So before we get into the analysis, a little bit of a background on Alzheimer's. So as many of you may know, this is the most common form of dementia. It's characterized by amyloid beta plaques and tau tangles. And so in the image on the right, you can see some normal, healthy-looking neurons. But when someone who's afflicted with Alzheimer's, they have these neurofibrillary tangles that occur within the neurons, as well as an accumulation of these amyloid plaques. So overall, this leads to the deterioration of the neurons and the decline in cognitive function. And so not only are the neurons the target for Alzheimer's disease, but microglia are also really important as well because these are the tissue resident macrophages of the central nervous system. And then a little bit about microglia and what research has been done in Alzheimer's. So there has been research done in a lot of mouse models where specifically people have been able to identify these subtypes called M1 and M2 microglia. These have been well characterized in AD pathology. So in the image on the right, you can see our M1 microglia. These tend to be pro-inflammatory. Which often lead to pathogenesis of other diseases. However, the opposite subtype, which is M2, these tend to be anti-inflammatory. So these could lead to the repair of the cells within the brain. More specifically, in amyloid mouse brains, other types of activated microglia phenotypes have been identified, such as this disease-associated microglia, which is likely to be involved in protective phagocytosis. And so the goal for today's webinar is I wanted to just kind of explore if I can find these subtypes within human brain and see if we can characterize them a little bit better. And for the data set that I was using in today's club is called Distinct Amyloid Beta and Tau Associated Microglia Profiles in Alzheimer's Disease. And if you'd like to access this data set, you can go to Talk to Data and search for the title here. And so I want to speak a little bit about how the authors have designed their experiment. So they have three experimental groups, CTR, CTR plus, and AD, which is Alzheimer's. And taken from two regions of the brain, the occipital temporal cortex and the occipital cortex. You can see here how in a control or a healthy brain, we don't see any amyloid beta plaques or tau tangles. But as we progress through the disease, this CTR plus is still considered a healthy control, but there are some accumulation of these amyloid beta plaques here, but there is no presence of tau tangles yet until we get to someone who's actually diagnosed with Alzheimer's. You can see a huge accumulation of those plaques and also the presence of tau tangles. And so today's analysis, I just wanted to compare these three experimental groups and try to characterize those microglia subtypes. All right, and so let's get into the first analysis, where the first thing we want to do is to just identify all of these cell types that are present within our data set. So switching over here to BBrowserX Pro, I've opened the data set here. And when you perform cell type prediction, the first thing you would want to do is you would want to perform a clustering analysis. So what this essentially does is it will group together your cells based on how similar their gene expression profiles are. So we have a lot of default parameters for you to just click run and for you to see the results here. So for example, in this previous result that I ran, here you can see my cells have been grouped into 15 different clusters. And each of these clusters may be of a different cell type, or they can be of the same cell type here. So for example, you can see here, down here in this orange and blue cluster, you can see three different colors going on. But these three different clusters could be of the same cell type since they're clustered together in that shape there. So once you've clustered together your cells, the next step that you'd want to perform here is the cell type prediction. So again, I mentioned how we have a lot of different tools built in for you to perform cell type prediction or cell type annotation. And for anyone who's new to BBrowserX Pro, I would recommend you start with this MetaReference tool here. Essentially, what this does is it utilizes our talk to data database, which is our database of single-cell RNA sequencing data, and it takes the marker genes from from that database, compares them to the marker genes within our data set to automatically annotate our cell types for us. So for the input metadata here, we want to define it as the clustering analysis that we just performed in the previous step. So for example, it would be the widen clustering here. And for the prediction type, you can also specify whether you want to predict by the major cell type or the subcell type. We'll choose the subcell type. And this last step here is really important. You would also want to customize the reference because of course we don't want to use the entire database of single-cell RNA sequencing data within our prediction because that database is composed of a lot of different cell types, a lot of different tissue types, conditions, and also suspension type as well. So what I'm essentially doing here is I'm going to add some filters to the database, filter out those studies which are not relevant, and only utilize the studies that are relevant in my prediction. For the tissue type, I know that my data comes from brain, so I will add brain to the filter. Okay, and then one thing that I failed to mention at the beginning is that this is single nuclei sequencing data. So you can also specify that within the filter of the cell type prediction here in suspension type. You can select nucleus. And so now if I can direct your attention to the right-hand side, you can see how we have a total of 14 studies related to brain that are sequenced with single nuclei. So if I click predict with filter here, and we view a past result, it's going to give you an overview of all of the predicted cell types. So right now what we're looking at is for cluster 1, you have a list of the predicted major cell types here, and then also a list of the subcell types. If we hover over one of these cell types here, you can see the list of core marker genes that call this macrophage a macrophage. And the percentage that you're seeing here on the left-hand side of each of the predicted cell types is the agreement rate. So the agreement rate you can interpret as the percentage of studies that quote unquote agree with this annotation of macrophages. So with an agreement rate of 50%, 50% of studies agree that cluster 1 is actually macrophages. And so more specifically, what this cell type prediction tool is doing is that it's getting this list of core markers here and comparing it to our list of marker genes on the right-hand side here. So you can see how for cluster 1, we have a list of the top 100 marker genes. And so when we overlap them and compare them to each other, then we can get some other types of metrics associated with how well that overlap is between our two lists of genes. So moving here, we can move to the details tab here. This is where you can get more information about what specific studies were used in the prediction. So if we hover over one of these studies here, for example, this one, you can see how the authors have annotated this particular cluster as a micro PMV, but basically the standardized cell type name is a myeloid leukocyte. You can also see the list of the author's markers and the overlap score, which is the number of genes that overlap between our two lists of genes that we're comparing. And then some other metrics such as a similarity score and a rank as well. And if we were to click on one of these genes here, this will navigate us to the next tab, which is the genes tab. And so now we're able to see the expression of that gene on our UMAP. You can see here how this Siglec1 gene is pretty specific to this cluster down here. But you can also look at the metrics involved. So we have the within cluster coverage percentage, the outside cluster coverage percentage, as well as the log2 fold change and the weighted log2 fold change values. Okay, and then finally, we have the studies tab here. So this is where you can get an overview of all of these studies that were used in the prediction of your cell types. If you click on the title here, you can read the paper. Otherwise, it also really gives you a great overview of the marker genes for those cell types here. And so after you've investigated all of these different metrics and you come over back here to the overview tab, you can also change the final annotation here. So for example, instead of annotating my cell types as a myeloid leukocyte, I can choose to label it as a macrophage instead. And so I've done that there, and I can click Save to Metadata. Okay. And in doing so, once it's saved to the metadata, now you can flip through the metadata here to visualize your cell types on your UMAP. Wait for it to load a little bit before I jump into how to customize this visualization here. Okay, and so now we see that some of the cell types that have been predicted here include our T cells, our astrocytes, our macrophages, and are also our microglial cells here. So now next, let's jump into the BioVinci portion. So for those of you who are not familiar, we've released BioVinci to some users. Not all users will have access to BioVinci, but for those of you who do, you will be able to see this export to BioVinci button here. BioVinci is our platform, which helps you customize a lot of the visualizations that you create on BBrowserX Pro or on SmartBulk, so that you're able to export it to BioVinci and kind of customize the figure a little bit. So if you want to change the font or if you want to change the colors, you can do so within BioVinci. Clicking here into export to BioVinci, now it's going to open up a new tab. Okay, and it's going to load the image here. Okay, and so once it's loaded into BioVinci, right now we are in what's known as the plotting mode. So if we want to enter the editing mode, we will edit the plot here using this button in the top right-hand corner. And now you can start to modify any of the things that you want to modify here. So for example, I want to modify the axis labels here, obviously UMAP2 and UMAP1. I can also change the font size for both of the axes. And let's change the title as well, cell types, and make the font a little bit bigger. Okay, so far it's looking pretty good. We also have the legend on the right-hand side here. But one highly requested feature that people have been asking for is they don't want to particularly see the legend, but they want to see the labels of the cell types overlaid on the UMAP itself. So instead of looking at the legend here, I can disable it using the toggle button here on the left-hand side, disable the legend, and now I can go over to the advanced tab here to enable the overlaid labels. So now you can see the labels overlaid on top of the UMAP like this. And you can also move around these labels here, and it will still point to that cell type. Okay, so this is looking pretty good so far. Now I can save my results, and I can show you how to export this image in a variety of high quality formats that we have. So clicking export here, the file types that you can export it as is as a PDF, SVG, PNG, or JPEG. I'll select the PNG. You can also change the resolution value here and also define whether you want a background color or no background. And let's go ahead and preview. Okay, so this is looking pretty good so far. Now I can export this and it will save it to my local computer here. Okay, and so this is what the graph is looking like. Okay, and so now let's go back to our slides here. So yeah, this type of visualization here that you're seeing on the slide was made with BioVinci. So anytime you see this little, go back to BBrowserX Pro here, anytime you're able to see this little export to BioVinci button, you can go ahead and export that chart to BioVinci and then customize it however you'd like. Okay, so next, since we were talking about a lot of visualizations on BioVinci, I also want to cover some other types of way you can visualize cell types on BBrowserX Pro. So one of the more common ways is, of course, through a heat map, where if you have a list of marker genes pertaining to a cell type, then you can list all of those marker genes and put them into a heat map here. Another way is, again, directly on the UMAP. So if you have a particular marker gene, for example, this IL7R gene, which will denote for our T cell population, then you can visualize that population on a UMAP like this. Or lastly, violin charts are also really popular in visualizing marker genes. So you can see here how I have two marker genes for our, I believe this is our astrocytes. So you can see here how the expression of these two genes is higher within these particular clusters. Okay, so yeah, this is just an overview or a summary of all of the different ways in which you can visualize cell types on the BBrowserX Pro. Okay, next, so once I've already identified my cell types, next I kind of want to see are any of the cell types kind of different across my experimental groups or across my conditions? So one great way of seeing this is by seeing if the cell type proportions are different between my different conditions. So again, I am comparing my three different conditions here. And you can see like a small pattern going on where our microglial cells, we see a slightly larger proportion in our Alzheimer's disease. However, this doesn't really conclude much. So what the authors have done instead in their paper is that they performed a chi-square test, which essentially allows you to associate two different types of variables together to see if they are statistically significantly associated with each other. And what they found is when they compare a healthy control with their Alzheimer's disease, that there is a high association of microglia cell types with AD pathology. So essentially what this is saying is that the expression profiles of the microglia cells are changing enough throughout our different conditions. So this would be interesting to explore even further. Okay. And so in this next slide here, that is exactly what I've done, where if we isolate our population of microglia cells, now we can look at our microglial cell subtypes. So I reperformed the clustering analysis just on my microglial population of cells. And then I performed marker gene analysis to find the top 10 marker genes for each of these different clusters or each of these different subtypes. And what I found here within this red cluster in particular is these top 10 marker genes are associated with a phagocytic or an activated type of microglia. And so this is one of another tool that I want to demonstrate here on BBrowserX Pro. So after we have isolated our microglia population here, again, I've performed the clustering already. We have this marker genes tool up here. And all you would need to do is provide it with the method of finding the marker gene analysis, the metadata, which again is going to be the clustering analysis that we performed in a previous step. And now if we click run, we are going to find the marker genes for all of the clusters here. And so in the results page, this is what it's going to look like, where for each cluster that is selected from the right-hand side, you get your list of marker genes on the left-hand side. So for example, it was cluster 4, our red cluster, where you can see the kind of enrichment of those phagocytic or activated microglia genes. You can also sort it by the log2 full change, so you can get the top 10 there. Okay, and so this is a really easy way for you to compare marker genes between your different clusters here. All right, and so now that we've identified that this red cluster here has a lot of genes that are enriched, that are associated with this phagocytic type of phenotype, another thing I can do is to look at the pathways that are enriched within that particular cluster. So on BBrowserX Pro, we allow you to perform pathway enrichment. And in particular, you can perform pathway enrichment using GC, also known as Gene Set Enrichment Analysis. And so that's the type of plot that we're looking at here. It's called a GC plot. And in Gene Set Enrichment Analysis, essentially what it does is it will tell you which pathways are enriched in a particular population of cells based on a calculated enrichment score. So each of these 3 colors here represent a different pathway. And as we see the enrichment score kind of climb up, the peak of the enrichment score is what we're really interested in. So if we see the peak of the enrichment score kind of fall to the left-hand side of the graph, as you can see here, then we can consider that pathway to be positively enriched in our population of cells. So again, basically I'm looking at, looking at this cluster, this red cluster here. And so we can see some of the pathways that are enriched within that red cluster include neutrophil degranulation, regulation of macroautophagy, which is interesting to see because, of course, we expected this to be a kind of activated or phagocytic type of phenotype. And then also signaling by PDGF. Okay, and so yeah, these are the types of some of the top pathways that are enriched within that activated or that phagocytic microglia subtype. Okay, and to summarize here, my colleague's going to open up the poll in just a few seconds here. So that while that one is circulating, I'll summarize some of the things that we've concluded here. So again, microglia subtypes in Alzheimer's disease have been identified in mouse models, but not really well characterized in human brain. So within this analysis, I was able to utilize the marker gene analysis and the pathway analysis to identify a microglia subtype which exhibits a phagocytic or an activated type of phenotype. There are also other tools which you can use on BBrowserX Pro to help you characterize these subtypes even further. For example, I can recommend using pseudotime or trajectory analysis, which allows you to track the gene expression change across different different trajectories or different pseudotimes. So I encourage you to use these tools to help further some of the findings that I found here. Okay. And so while the poll is still circulating, I will also open the floor to any questions. So go ahead and submit any questions to the Q&A, and then I'll do my best to answer them. And then also related to the poll, I do want to share a little bit about what we will be working on for this month. So this month we are going to be making an enhancement to the cell type prediction feature. So I just kind of want to know, for those of you who are using any type of cell type annotation tool, what methods you do use. And we can maybe research them and see if we can implement them into our roadmap here. And then feel free to also leave any feedback or feature requests in the poll as well. Okay, so we do have a question here. One person asks, so you're using the agreement percentage from other marker papers for cell type calling. 49% is good enough for calling that cluster as macrophage as explained. What would be the minimum percentage to call a cell type confidently? So yeah, this is actually a very, very common question whenever I present the cell type prediction feature. So while I can't say there is like a definite threshold for calling a cell type confidently, but from my perspective, let me open up the results page. Okay, from my perspective, it's kind of just a mix of not only just looking at the agreement right here, but also if you're someone who is kind of more of an expert on some of these cell types, you can kind of investigate these core markers here, and then see if they match pretty well with your list of marker genes here. But again, you can also look at some of the other metrics. For example, the overlap score, the similarity score, or the rank. So for example, to utilize like the rank metric, if we see our list of core markers here, some of the top markers for the macrophage include C1QC, CD163, right? And if we were to compare our list of marker genes on the right-hand side, and we see that CD163 and C1QC also rank pretty highly on the top of our list, then we can be pretty confident that perhaps this cluster 1 is our macrophage. Because not only does the number of genes that overlap between our two lists of genes matters, but it also matters the rank of those genes within those lists. And so this is how the genes tab here can come in pretty handy, where you're able to look at the rank of these particular genes that you're interested in, and also the coverage here. So for example, if you see a lot of coverage outside of this particular cluster, then you can conclude that that gene is not really specific to that cluster. It's kind of expressed elsewhere within other clusters, so maybe it's not a good marker gene to use for that one cluster. Okay, so I hope that clarified your question a little bit there. Okay, so if there are no other questions from anyone else, thank you so much, guys, for joining today's Single Cell and Exploration Club. Next week, we'll be hosting another webinar, but during Asia-Europe time. So tune into that, and thank you for joining. Okay, bye, everyone.